to say thank you to all the sponsors. Like Tom said, it's a nonprofit, so the sponsors are really important to have these types of events and bring all the automation specialists all together. So today, what I will be presenting, I'm going to go through a journey with you, my journey, and you'll see that the evolution and where I am today in the clinical space, right? So that's the title, right? So high throughput drug discovery to automated clinical research. And I'm going to give you my perspective from a regulated standpoint. So here's the agenda. Have to present who's AltaSciences, my experience in drug discovery, clinical research, and then I'll give you my future perspective. So AltaSciences, we're a CRO, so contract research organization. We are based mainly in North America, Canada, and US. The headquarters is in Laval, right here, and we have a clinical site in Montreal where we recruit patients. And I'm on the lab side where we test all the samples from the clinical to support clinical research that we are contracted for by the pharmaceutical industry. So we do offer full service CRO, CDMO. On my end, it's a bioanalysis. And we support anyone, pharma, biotech, academia, anyone that has a drug that they would like to bring to the market. And so basically, we go through a contract together and go through this study protocol. So just this is an interesting fact, right? AltaSciences, born and raised in Montreal. It used to be Algorithm Pharma. Some people might remember this name, and it was actually founded by some researcher at the Montreal University, Dr. Gilbert Caillé, who's the founder, and with Marc Lefebvre, Jean Spinard, and Louis Caillé was the president at that time. So that was in 2000, roughly 2008, 2007, 1992, sorry. And then it became AltaSciences in 2019. And with over time, Algorithm was basically a software to support clinical research. And then they realized pretty quickly that they needed also to analyze those samples. And this is where CRO services, bioanalysis, and then with time, we acquired additional companies to do preclinical research where we have in vivo facilities. And so today, AltaSciences, so like I've mentioned, we have multiple sites in Canada and in the US. You can see on the West Coast, middle of the US and East Coast, where pretty much clinical facility is linked with a bioanalytical facility. You can see in Canada, so we have Laval, like I've mentioned, it's bioanalytical bioanalysis, and the Montreal clinic. We also have a presence in Europe. AltaSciences is growing. We've been growing for the past, well, you saw since 1992, but every year we're growing, we're acquiring additional labs. And so obviously Europe is probably our next step. So this is the total solution. Obviously here in different colors, you have all the different sites, preclinical. But if you look at the full spectrum of drug discovery, from discovery, non-clinical to clinical, if you focus on the light green, maybe I'll use a pointer here just for the light green. This is Bioanalytical, so this is my shop here. So we do cover, we do a little bit of discovery, but the majority is non-clinical work where we get samples from animals for testing, and then we support the phase 1, phase 2, and phase 3 up to phase 4, up to basically we generate a report for the FDA or Health Canada or etc. So this is our lab. So what do we do in our lab? So we do have the Laval site is the biggest site where we have about 300 employees, and you can see the comparison in Seattle and Columbia. So this is really lab science or bioanalysis. So we do have LCMS, ligand binding assays, ELISA, MSDs, etc., flow cytometry, immunogenicity, biomarkers, cell-based assays, ELISpot. So we do cover the full spectrum of what is required for clinical research. Now, all right, enough about the corporate aspect. Let's focus on my experience and what's really interesting, and that's why putting these slides together, I thought it was very interesting because I've just celebrated my 25th year as a professional career this past April. And when I going through these slides, and the message I wanted to bring is I thought it was very interesting to see the amount of automation that was present in my career. So as I graduated from the University of Montreal with in molecular biology, I started immediately at Merck Frost. I don't know, maybe some people don't know Merck Frost. It's part of Merck. We had a research facility in the West Island, and that closed down in 2010. And this is where I was transferred to the US, where I was the director of assay operation there in the US. And that's a sad part of my career, but you know, we can't control everything. I did a little moment in sales with Beckman Coulter. And then I realized pretty quickly that my space is and my forte is in the lab with automation. So I joined the NRC as a lab automation operation manager. And then in 2021, I had an opportunity at AltaSciences where they were asking me, well, maybe you should join AltaSciences to help us build automation within our clinical research. And so today I'm a senior director of bioanalytical operation, but my expertise is really process improvement, lab automation, miniaturizing assay, sample management. So I'm going to go through each step of my career. And basically, I started just like any one of you, right? As a bench scientist. And when I started at Merck Frost in drug discovery, I was basically supporting all the medchem programs. And I was doing everything by hand, right? Using pipettes, multi-channel pipettes, electronic pipettes. And I was receiving all these samples in vials, you know, just storing manually into the freezers. And then on a weekly basis, they were telling me, well, we have all these samples, you need to test those. Can you do that for us? Yeah, sure. So that was the first job. So that's fine. But very quickly, within the year, you know, they said, okay, you're increasing throughput, you're very productive manually, but hey, we have a liquid handler right here in the corner. Do you think you could do something with this? You know, young guy coming out of university technology, and like, yeah, for sure, you know. So here's the manual, learn it and develop and automate this assay for us. And so that's basically how I started in automation. And then not long after that, and I was asked to move into the high throughput facility department, where at that time we had a CRS robotic integrated platform. And I'm sure a lot of people will have some memories here. So CRS robotics, which is now Thermo. It was the main workhorse for us. And you can see the type of instruments that were integrated with this platform. And it was running on Polara, which was a static scheduler, right? Today, everybody, I think, has a dynamic. I don't think we hear about static scheduling anymore, but very reliable, right? And you can see the instrument. This was a robot on a rail. And we had carousel. Workhorse in terms of liquid handling was the Biomech FX dual arm. We had a Spectromax Gemini, very basic, sorbents, fluorescence, plate washer, and Embla. Oh my God, that gave me a lot of headaches. But you'll see in the next slide what we were able to do with all these, with this platform. So these are just two examples, but the main goal of this platform was to support the MedChem program. And obviously doing everything by hand. So I'm showing here on the left, cathepsin K for osteoporosis. It's a protease, and we manually, we needed two scientists to basically test all the compounds, the small molecules that were produced by the medchems on a weekly basis, right, in 96 format. So again, with this platform, we wanted to increase the throughput. They said, okay, so we'll optimize that in 384. So bottom line, at the end, we were able to screen all the samples. You can see titration curve at the bottom, 10 points titration curve. But we had, we were basically screening cathepsin K, but also all the other counter screen like CAT-B, CAT-S. So 6 different enzymes on a weekly basis. So we could generate full IC50 curve for about 384 samples every week. This was very useful. And obviously I was running this, I was a single scientist running this. But at the same time, we said, well, maybe we should automate even more. We should automate data analysis and the reporting. And so with the internal IT, we developed what we call MAPS, for Montreal Automated Plating System, which you have a snapshot here, where you could order your samples and being put in the plate for screening. So with that, you know, at the end, it led to the discovery of Odanakatib, and which didn't make it to market at the end, but it was pretty interesting. The other aspect was HTS, right? So on the right side, PGE synthase, which is for inflammation, it's in the same category as Vioxx, which was drawn from the market. And so obviously at the time we had to perform a screen for novel entities. And this was an enzymatic assay with a very unstable substrate, PGH2. But at the end, since they couldn't run this in our high throughput facility in the US, they asked us to screen the collection because it was a very complex assay. So using the same platform, we performed an HTS screen. And at the time, it took us 6 months to screen 350,000 compounds. Because of the assay, it was complex, but we transformed that assay from 96 to 384. But we were successful by identifying 3 novel entities, but at the end, program was canceled because of the Vioxx issue. That being said, very reliable platform. You know, we were able to do what we needed to do. Next step was, okay, now we need to go even faster. So let's go in 1536, 3, 4, 5, 6 well plate. I don't know if anyone here has ever seen a 3, 4, 5, 6 well plate, but Aurora is a microplate. It used to be Aurora Biotechnologies. And they were also producing not just plates, but they were producing very efficient instrument like the Bioraptor, which was revived recently. And that was a high throughput dispenser. You could dispense 8 reagents at the same time in a 3, 4, 5, 6 well plate within 2 minutes. Amazing. So it is back on the market. So if you want to look at it, very interesting. And they also had this topology compensated plate reader. Amazing instrument again. But so to go to the ultra high throughput screening, right? We had this collaboration with High-Res today. It's called High-Res. At the time it was High Resolution Biosolution. And what was super innovative in this platform was being able to have docking station and rolling carts. So here I'm talking about in 2008, right? So you can see here Stably robots, Biomech NX, the standard dispenser, washer, but we also had a Vulux. Vulux was a imager. It was humongous. You can see the size here. Has a huge camera in order to be able to read 3, 4, 5, 6 well plate. And you have a snapshot here of a 3, 4, 5, 6 well plate in fluorescence mode. So you can see the signal is very, very clear. But that was a combination of technology that were coming out, giving us the opportunity at that time to go into 1536 and 3, 4, 5, 6 well plate. Our facility down in the US at that point was able to screen 2 million compounds within a few days, 3, 4 days in this format, which is today, in my mind, is standard almost. And we had the Echo, which at the time was lab site. Again, revolution, right? Today, it's very, very common to have an acoustic jet dispenser. So then I moved to the US, that's in 2010. And then very similar role where we were supporting the MedChem programs, but just more expanded, right? So we're supporting about 10 to 15 MedChem programs at the same time. And then at this moment, there was a lot of molecule coming in. So we had a fully integrated sample management process where, chemists were bringing in their samples. We were basically standardized on the Hamilton liquid handlers, and basically all the samples were going into an automated store from Brooks, which is today Azenta, and we were preparing all the plates. And then we had multiple screening platforms, Velocity 11, which is Agilent today, the Bravo, the bench cell that you hear. So this were all integrated together. Thermo DM4, really cool little robot, just like swing back and forth. Each instrument had their little robot that was attached to them, still on the rail. And then we had multiple high-res platform, very efficient. And at the time, the main difference here is that we were also the central lab for the hit characterization in 1536. Once the main screen and the hits were identified in our HCS facility, they were sending us the Kanban, and we could use these platforms for hit confirmation. So, all right. During that time, this is where my career went to another direction. CRO outsourcing became really popular. That's in between 2010, 2012, while I was in the US. And basically what Merck was telling us is, you need to show everything that you're doing, and they'll be fully integrated with us. What did that mean? They had the same instrument, the same software. They were doing sample management for us. They were doing high throughput screening for us. And the main company at the time was Wuxi, and they were based in China. So we were successful. But that being said, this is where I started to be exposed to CRO. And so at that point, well, I said, well, maybe I should come back to Canada, and maybe I should help my Canadian colleagues to do drug discovery. The main purpose at the time was cost reduction. That was the main goal, right? For the pharma industry, was to reduce costs. So instead of joining an actual CRO, I decided to join NRC, which is the Canadian RTO, Research Technology provider. And at that time, I was a lab automation operation manager. And you can see here, you can appreciate the type of instrument that we had in terms of, so we had multiple liquid handlers, the TCAN, the Hamiltons, we had the Cynomics labeler, the Biomex, and we also had purchased the Ritisoft platform that was in maybe 2015, 2016, if I'm not mistaken. That being said, right, it's always the same type of instrument, right, that we've been using. But what were we doing with these platforms? So we do have Eve here. That's the expert in protein production at the NRC. So at the time, we were producing antibodies, and so we were using the Hamilton platform for 96 choke protein production. Again, the goal was to increase throughput, decrease the costs, save time, decrease manual labor. But what did we do? Once you produce your protein, so this was in 96 format, then we adapted the TCAN Evo using robocolumns and centrifuges. So robocolumns, which are packed with affinity resin, that you can purify all your antibody, and using centrifuge for basically do buffer exchange. So again, same principle, right? Being able to increase the throughput and decrease costs. Then we were using the biomech effects for sample management, with the 2D barcoded tube racks. And then at the end, basically characterizing these samples using the stunner and also the molecular device high content screening. This was a cell-based assay. We also had a biotech for ELISA to characterize all of our antibodies. So going from ultra high throughput integrated platform with workstations, right? So a bit less automation in my life, but still very useful and impactful. Then I decided to join AltaSciences as a senior director, sample management and automation. The main reason, so basically they want to implement automation in a clinical space, regulated space, right? There's a lot of challenges that comes with this. But I said, you know what, this is a challenge. So let me jump into this space and, see where it goes. So I'm not going to go through all the challenges because we could talk for hours about challenges in the regulated space, right? But basically, we need to make sure we follow all the regulations, Health Canada, FDA, AMA, and Visa, and basically follow the guiding principle of GCP and ALCOA, right? Which is ALCOA. I don't know who in this room is working in a regulated space, but ALCOA basically tells you that you need to track everything from who did it, what time, and it cannot be at 11 o'clock if you did it at 10:30. You need to write everything down, and you need to make sure that all your instruments, all your software is validated, right? What does it mean, validation? It means that if the instrument is going to pipette from A to B, you need to confirm it's going to pipette from A to B and document all of this. If your software has different roles, you need to confirm that these roles are actually true, and you need to build test case in order to make sure that, all the permission that you gave that person is true and it's reliable over time, right? And what's really important is the audit trail. What we hear a lot, and that's why I've put at the bottom vendor approach. Many of the vendors, they come and they say, yeah, we have an audit trail. Yeah, we are 21 CFR Part 11. Okay, yeah, we do have roles and permission. Yeah, but it's not just that, right? What's in your audit trail, right? If your audit trail doesn't show the user in there, no, there's a gap. We need to document that, right? So I'm giving you really just a small snapshot of a regulated space, but there's a lot of challenge. And when our worst nightmare is a mistake, if someone does a mistake, if you have a bad data, because it's not like you ran out of samples, you say, I'm going to get another sample. It's human clinical samples, right? You ran out of that. Oh, there's no more. Then the auditor comes in. Yes. Yeah. The auditor comes in and asks you, all right, what happened to that sample? Oh, we had a failed instrument. Okay. Did you document that? Yeah. Who was running? Who was the person qualified? You have his pedigree, his CV. Show me that this person was qualified to run this, right? Do you have a copy of his diploma? That's the level of what an auditor comes into when, so you can appreciate the amount of challenge, right, that we can have in our environment, right? The other challenge was basically, when I got in, everything was manual, paper-based. So I'm trying to get into that digitizing our process and having a good audit trail, right? So these are my challenge. Timelines, the sponsor, they tell you, nope, this is the protocol. You need to vortex. Okay, it's hard to vortex on the liquid handler, right? We can do it another way. No, you have to vortex, right? So there's a lot of constraint like that we go through, which makes my day exciting and challenging at the same time. So what do we have at AltaScience? You'll see very basic instruments, right? We have a Cynomics. When I joined, basically they had Kingfishers, they had biotech reagent dispenser and washer. So now we had also some Starlets, different configuration, and now we have some Integras. And so what do we do with those? This is an example where we have a Kingfisher. And once you've validated this instrument, then it becomes much easier to say, I'm going to buy a second one, right? So if you look at this Kingfisher, you know, it's a simple automated platform, right? So we have eight of them, right? So we can produce more, right? And then we have a lot of mass specs, 2 of those, and what, 16s here? Yeah, 16. But this process here, you know, is manual. So we're working on that to automate the sample prep. Then we'll use a Kingfisher. So this is a simple example, right? How we can implement automation in the clinical space. What we're really working on is this high throughput automated clinical ELISA as an example, where we would use our Hamiltons to do standard in QC, sample dilution, connected with our LIMS, and then use the reagent dispensers and washers to perform that. Read this, automate our data analysis, our reporting, and also our KPIs. One critical aspect in the CRO world, budget, revenue, money is super important. So we cannot have an instrument that's accumulating dust in the corner. We need to prove that we're using this instrument, right? So KPIs, key performance indicator, really important. So we're trying to automate that. Actually, we are automating it, just not to the level where we want to be, right, DaVinci? All right, so if I reflect on my career, right, and if you look at this graph, so this is what I was trying to highlight. So automation on the Y scale, time from 25 years. All right, I've learned. Merck gave me a lot of opportunity, a lot of automation around surrounding me. Then I joined NRC, right? Yeah, level of automation decrease, but still very valuable, right? Then join AltaScience, a little bit of automation. And where am I right now? I'm about here. So what's the next step at AltaScience and in my career? Will it go back to here? I don't know. Will it go kind of settle in the middle here? I don't know. We need to determine how much automation we need in our space. And it's all based on the regulation, right? So what am I seeing? How much time do I have left? 1 minute, should be okay. All right. I went to SLAS recently. I don't know who was there. Went to SLAS, was 5 years last time I went. Wow. We're in the new era, right? AI is coming. I know AI, some of you are using it in drug discovery, in academia, right? But the pharma industry, they have high throughput screening. They have high throughput chemistry, right? They're using AI. So there's more and more samples in drug discovery coming to the clinical phase, and good molecules also, right? So what's going to happen? This is going to put a lot of pressure on companies like us in the clinical research, right? To have the throughput to test all these molecules and fast and at a lower cost. I think we all have the same mandate, right? But that's going to put a lot of pressure. So if I take my workflow here, what's going to be the future state, right? So I've mentioned integrating all these instruments as a process, right? As a process. Where are we going? Am I just going to duplicate everything, right? Just have multiple instances of the same instrument? That's one approach if you want to increase, but I still need a lot of labor, right? And I know Hamilton seeing this, they would be very happy, but am I going to use cobots, right? To integrate all these instruments together? Might not be enough. Might need multiple platforms. Autonomous mobile robot. That would be interesting, right? Or humanoids. we're seeing it. It's coming, right? So just want to finish off on this last image. AI, we are in this new era. It is a game changer right now. But how do we implement that in our lab, but in a regulated lab like myself, where everything needs to be validated? How do you validate AI when your large language model are doing something that you're not quite sure what it's doing, but it's doing something, right? But we are implementing AI, and we want to implement AI and then have equipment attached to that to increase our throughput, reduce costs, accelerate timeline for drug discovery and drug development, actually. On our side is drug development. So thank you so much, and I'll take any questions.
Yeah, time for one quick question from the other. Obviously, Fred can be around for the rest of the day, so you can always catch up with him for that too. Anyone?
Marlon, question regarding using automation in a GCP environment. What were the biggest challenges you had transitioning to that?
The biggest challenge?
Yeah.
Number one is adoption.
Okay.
Right? That's the biggest challenge. But also, you know, you have to mind. Yeah. Yeah. But also, you know, when I got there, manual processes, right? In a rigid format, built in with SOPs, every little changes that you do, you need to update your SOP, you need to revalidate everything. So changing a manual process, going to automation, you have to go piecemeal, right? One step at a time. So that's the biggest challenge. And then obviously the regulation.